and cell cycle. Mitosis is really similar to meiosis, except in that it has a single division rather than two divisions. And when we think about the purpose of mitosis versus meiosis, that makes sense then. Because meiosis, we want to reduce the chromosome number to form gametes, whereas mitosis, it's about replacing cells, right? Or growing new cells. In other words, we want to make exact copies of the cell that we're working with. And so by having just the single division, we end up with the exact same chromosome number, and we're not reducing that down to form gametes. And so that's how that first cell division ties in there. We can see some images here that show us some of that, examples of that under a microscope, right? So we would have just where we have the nucleus still intact, starting to break down, and we have the chromosomes condensing. D on this slideshow would be a great example of that. So that'd be prophase. And then the next step, just like in meiosis, is a metaphase. And in metaphase, the chromosomes align at the center of the cell. And so we would see that alignment here. And then we would have anaphase, where the chromosomes start to separate. We can actually see, that process kind of carrying out here, here, and here, right? We can kind of see different stages of that. We can see kind of the ending stages here. And then we start to have the cells, the nucleus reform, and we have the cell starting to divide in a plant cell like this. We'd call that the cell plate forming right there. And we have two distinct nuclei forming there. So prophase, metaphase, anaphase, telophase is kind of the same steps we're seeing. And again, the purpose of this is to create exact copies of the cells rather than gametes, so that's going to be one of the key differences between this and meiosis. Mitosis is directly tied to this process known as the cell cycle, and that's what this image is showing us here. If we had just seen a cell go through mitosis, it would have just ended this portion of the cell cycle right there, okay? And it would enter into a stage of the cell cycle we call G1. That's this segment here. Now, G1, S, and G2 are all part of what we call interphase, like the phase between mitosis stages, right? But we divide up that interphase into these three sections, G1 being the first one here. So we're taking this cell that has just divided. We need it to get bigger. So that's one of the big things that will happen. We need to get more organelles, like mitochondria or chloroplasts. All sorts of organelles really are going to duplicate here. And probably most importantly, the cell is just going to do its job. And that's really where it spends most of its time in G1, is just doing its function as a cell. When it gets to the end of G1, it's reaching a time when it's preparing to divide again. If it proceeds past G1 into S, that cell is going to divide. And so there's a really key G1 checkpoint here. And this image kind of shows that for us here. The G1 checkpoint says, I need to divide. I have the things needed to divide. Is this cell big enough to divide? Do I have the nutrients needed to divide? That's what the G1 checkpoint is for. It's also going to check and make sure, is the DNA mutated? And if the DNA is mutated, it can actually do some repair function at this point. But if it's too damaged, that cell will will not proceed through cell cycle. And you can think about why, right? If we had mutations or damage to a cell, we wouldn't want to make copies of that cell. And so that's what the G1 checkpoint helps us do. And if it is not healthy enough to go through, it can either delay to see if it can fix some of these factors that it needs or nutrients that it needs, or it can go through, if it's too damaged, it can go through apoptosis. And apoptosis is where that cell will be destroyed. It's kind of a self-destruct button for the cell. to prevent damage from being inherited by future cells. And so that's what the stop G1 checkpoint is for. There's another way out of G1, and I don't know if it's necessarily out of G1, but it's going into a different phase that's not depicted here, and that's called G naught. In G naught, it would be a 0, and where the 1 would be here, right? G naught is when a cell is no longer going to go through cell division. Essentially, it will just perform its job and not continue to go through the cell cycle. So it's kind of pulled out of there. Sometimes you'll see it called cell retirement. I don't like that term because it implies that the cell's not doing its job. It still keeps doing its job. So cell retirement, just be careful with, like, if it helps you remember that it's not going to go through the cell cycle anymore, great. If it gives you the idea that it's not doing its job, that's not great. So there's lots of cells that go into G naught for humans. One is our muscle cells. So your muscle cells, it might surprise you, do not go through cell cycle. A lot of people will think, oh, that's probably how your muscles get bigger. There's mitosis happening, the cell cycle's going on, and I get more cells when I work out, and that's why my muscles get larger, right? There must be more cells there. It's actually not what's happening. What's happening instead is there's these fibers in your muscle cells called myosin and actin. They're protein fibers, and they wrap around each other. And when I believe it's the actin coils up, might be the myosin. When it coils up, it pulls the fibers of your muscle cells together, right? It shortens them, and that's what allows contraction of your muscles. Well, what you're doing when you work out is you're slowly breaking down those fibers to some degree. And that's how you get some of that soreness that happens, is from that breaking down of these fibers. Well, when those fibers heal, they heal back thicker. And so each muscle cell is actually getting thicker. You're not gaining muscle cells. And because we can't do mitosis to those muscle cells, because they're in G naught, those cells actually won't repair damage. This is why it's such a problem. Your heart is a muscle, right? If your muscle cells of your heart die, they're not going to go through mitosis to repair that damage. So that's why it's such a problem when muscle cells of your heart die, because they won't be repaired, right? That's going to be pretty much permanent damage almost all the time when that happens. Next, we have nerve cells that don't go typically, they'll go into G naught and they'll jump out of the cell cycle. Think about nerve damage, right? That is so difficult to heal, injuries to nervous system, because again, the body doesn't do it on its own through mitosis. And then brain cells is another one, right? And so those muscle cells, brain cells, and nerve cells are all going to be really difficult for healing to take place, because the mitosis is not normally happening for them. And as a result, we don't have that natural repair place where we can replace those damaged cells if they have died or become too weakened. So that is G naught. Assuming a cell doesn't enter G naught, it passes G1 checkpoint, it will go into what we call S phase. S stands for synthesis. This is when we're going to take our DNA and copy every single chromosome in that process. So that's what's happening here. We're getting those 2 copies of the DNA. And that takes in humans, I think I remember it takes about 24 hours for that to take place, right? This DNA replication process. We'll learn more about it in a future lesson, but just know that it takes place in this S phase here. Once we get done with S phase, we'll enter into G2. G2 is just kind of a period where not much is happening. Some energy is being acquired, some organelles might be copied in this stage, but there's not much happening in G2 before we reach mitosis. And we'll reach at that point a G2 checkpoint. G2 checkpoint makes sure that the cell is, again, large enough to divide. We've gotten bigger because we're going to have to split in 2. That's going to make that cell much, much smaller. And then we need to make sure that the DNA was completely replicated. You don't want to start dividing before that DNA replication process is complete, because then the daughter cells won't get a copy of the DNA, and then it will go. If it passes G2 checkpoint, it will continue on and go through those phases of mitosis that we talked about, and we'll get 2 new cells. All right. So one of the key things to understand that relates to cell cycle and mitosis is when it goes wrong. When there is uncontrolled cell cycle, just growth happening constantly, that is going to form a tumor. And tumors are cancer, right? And as we've come to learn more about cells and why they divide, there's 3 things that kind of stop cell division from happening and should, in theory, stop tumors from forming. The first is what we call contact inhibition. Cells don't like to be crowded. And as a result, mitosis will be shut down and the cell cycle will stop for a period if there is contact inhibition, if there's crowded cells together. We also have what we call anchorage dependence. We need other cells or other structures of the body for those cells to attach to. That anchorage is what allows, again, the factors to be present for mitosis to be triggered. So we have contact inhibition and anchorage dependence. And the last thing that keeps our cells from undergoing cell cycle uncontrolled is what we call telomere shortening. Now we have our chromosomes, right? Our lengths of DNA. On the ends of those chromosomes, if you remember, we talked about this in our human evolution lesson. We talked about chimpanzee chromosomes and human chromosomes. The ends of those chromosomes are called telomeres. They're very specific sequences of DNA that don't have genes in them. They're just protective caps that go on the ends of our chromosomes. When DNA replication occurs, it's not completely done. And so every time your cells go through DNA replication, a little bit is left off, and we call that telomere shortening. Now, it's not a problem for your cells when you're younger because there's plenty of telomere to work through. But as you age, there's enough DNA replication that has happened that you could work your way theoretically all the way through those telomeres and start getting into sequence of the DNA that actually codes for something that is actually a gene. And so that is the third control mechanism that stops that. But cancer cells have overcome each of these, right? They don't exhibit contact inhibition. For some reason, the signals that would stop mitosis from contact inhibition have been shut off in tumor cells. They also don't require an anchor. They will divide without having something to attach to, which can be problematic, of course. And the last thing is telomere shortening. Somehow they've activated a gene that rebuilds the telomeres of our chromosomes. That gene is, that enzyme that does that is telomerase that's formed from that DNA. And so we get these 3 things that should stop the cancer cells from dividing, but don't. It's thought that much of that is triggered by what we call tumor suppressor genes. Those are genes that stop tumors from forming. And proto-oncogenes, these are genes that encourage cell division to happen. That between these, and there's lots of there's over 100 genes involved in this, right? I think 140, 150, somewhere around there. These genes, if they are functioning properly, will regulate the cell cycle. But if we have mutation happening to either the tumor suppressor or the proto-oncogenes, we get those parts of the cell cycle. The more and more of those 140, 150 genes that get mutated, the more likely we can have cancer develop. And so if you're born because your parents or grandparents had mutations to those tumor suppressor or proto-oncogenes, you have a increased risk of cancer happening because you are relying on just a few of those genes instead of that whole protective suite of genes stopping cancer from forming. So that's how someone can be predisposed to cancer, because they have already had mutations. They inherited those mutations, which means if those mutations happen to those genes that can trigger that cancer forming.